Our protocol uses a non-invasive brain modulation technique called Transcranial direct current stimulation, or tDCS, to reduce online gaming use.
tDCS is simple, safe, and inexpensive to administer compared to other brain stimulation techniques. We also use neuroimaging to assess brain changes related to stimulation.
After recruiting both online game playing and non-online game playing individuals, explain to each participant the aim of the study, the main experimental procedures, and any potential risks associated with participating in the study. After answering any questions, obtain written consent. Have each participant fill out questionnaires, such as the Internet Addiction Test and Brief Self-Control Scale, and ask the participants to report the average weekly hours spent playing games. To perform an FDG PET scan, inject each participant with 185 to 222 megabecquerels of FDG, and have the participants rest for 45 minutes in a supine position in a dark and quiet room with their eyes closed to allow FDG uptake. At the end of the uptake period, use a PET-CT scanner to acquire transaxial emission images and CT images in about 15 minutes, applying standard attenuation correction, filtering, and reconstruction techniques. Within a week after the baseline assessment, seat the